Hello, everyone. Welcome to the New Jersey Mycological Association Taxonomy Tuesday for January 27th, 2026. My name is Lila. I'll be the host this evening, and we're starting off tonight with a short presentation by our member Dorothy Smullen, who is also the chairwoman of our herbarium. So I'd like to turn the meeting over to Dorothy to give us a presentation about our herbarium, and then we will move into our normal discussion of member finds. So, Dorothy.
Is it up?
Yeah, we see like your whole desktop.
Oh, oh,
So,
just the whole desktop. Okay,
yeah,
hold on.
there we go.
Okay, great. Um, well, I was quite surprised to get an invitation to be a speaker. Our herbarium currently is housed in Rutgers Chrysler Herbarium, and they had a 100-year Jubilee Symposium on Friday, October 24th of last year. And I was very excited to be asked to speak. There were so many people that were speaking, lots of PhDs. Everybody was limited to just 15 minutes. So, but here's the beginning of that presentation I gave. It's called The History and Fun Facts of the Raymond M. Fatto Eugene H. Varney Herbarium. And that patch, by the way, was created by Rhoda Roper, one of our members. We don't see her much anymore, uh, but I think she got a life membership by winning that contest. So, how did we get started? Well, in 1971, an article in the Dover Advance by Hiram Korn wanted to find people interested in joining a mushroom hunters group. And people met and they formed for a couple of years the Lakeland Mycology Club. They they had displays at several local events up in North Jersey. But by 1975, the group became known as the New Jersey Mycological Association, and we are the only mushroom club in New Jersey for the whole state. Actually, we now have the Rutgers Mushroom Club, but other states, of course, many of you know, uh, have more than one or many. New York State has a whole ton of mycological clubs. I joined in 1975, uh, and there were monthly winter meetings, and there was a newsletter every month, and forays spring through fall. And I don't know what day of the week this was. Maybe it was a Tuesday or maybe a Monday. The first taxonomy session met after one of our forays, in, as you can see, in September. People wanted to get together to try to identify the species we collected on the weekend. And of course, some of us had microscopes to bring, and we brought our books. This is a really cute cartoon by Gertrude Espenscheid. She was a wonderful member. She was also an artist that did drawings for the Golden Guide books for children. But she, if you look in the old newsletters, you can see some of her cartoon work. So after that first meeting, Bob Peabody and I were interested in trying to save some of our identified dried specimens with, of course, the name, the date, location, and the identifier. There's a picture on the left of Bob at one of our Fungus Fest events. He was a great speaker, and he did a foolproof 4 demonstration for many, many years. And of course, he was president at the time. On the right, we see me, and in the middle, Jeff Kibbe and Bob, and that picture was taken at 1985 Nymph Oneonta. So we decided not to just collect some of our specimens, and Bob actually had a whole ton of different sized boxes, and we were using boxes. But this is the first page of our accession booklet. So the first specimen we put in was Agaricus abruptibulbus. It was quite large. And that was the date, January 31st, 1978. And there are now over 3,400 specimens. Since 2019, most have MO numbers, which most of you know, Mushroom Observer, but many have also been sequenced for DNA since that time. So our club did a workshop which was led by Igor and prepared by Igor in August of 2023, which we did DNA sampling at the Pinelands Preserve. We see Jason Hofstad in the center, Igor on the left, Liz DeCicco, I think her last name is, and, Igor went through a lot of preparation to do this. We only had maybe, as Igor said, 50% of the specimens, giving up their names, but still that's great. But back to our history. In the May of 1978, we actually received an invitation to house our herbarium at Cook College from Dr. Roger Locandro. He was an associate dean at Rutgers. And at the time, that's a picture of Martin Hall on the right, and you see Dr. Locandro on the left. Martin Hall is now, I believe, really used for administration, but at the time, it's also quite famous for, I believe, it was discovered streptomycin, maybe one of the antibiotics was discovered there. But we met in room 310, which was adjoining the attic, where the Cook College had their pathology specimens. We basically only had a couple of old filing cabinets, not true herbarium cabinets. But next we see sometimes we met at Martin Hall and up on the 3rd floor, and sometimes we met in other places, including, again, my dining room where I'm sitting right now, because that's where that first meeting took place of our taxonomy group. Club member Rod Tullis spoke to us about Amanita, and most of you have hopefully have been to his website. I know there's a problem connecting with it now, but he gave us a talk at Martin Hall. But by the late 1980s, the club had purchased 5 herbarium cabinets, where you see them on the screen. Official cabinets. Another club member, Ray Fado, he became an expert in the genus Russula. He became an East Coast authority on the genus, and he and Jeff Kibbe who now lives in London, wrote together the keys to the species of Russula in the Northeast, North America. So you can see the image of the first page of that booklet. I believe that we club might even have some that are available for sale. It was reprinted. And you
Yes.
see Ray and Jeff at the new NJMA Club picnic. I've got something blocking my screen so that I can't see what the date
Yeah,
was.
there's a hide on that. Can you click the hide to the right? Yeah, there you go. Right there.
Would I be able to see the stop sharing? Okay.
Yeah, we'll figure it out.
Okay. 1988. So it was quite a wonderful code system that Ray developed. 7 characteristics, and what you did, you looked at your unknown specimen and you picked the correct letter of these 7 characteristics, and you got a code. This is a multi-access key. It may have been a mistake to put peeling cuticle down for the second characteristic, but nevertheless, it was great, and it let people see the wide range of colors that you get from the spore deposits. And once you got your code, it would lead into the booklet to smaller keys, which were arranged in a dichotomous pattern. So over 1,000 of Ray's specimens are now found and digitized at the New York Botanical Garden. And here's one, which is Russula sylvicola, and too small to see where it was collected, but he took wonderful notes and diagrams of the spore patterns and so on. Ray, and here he's pictured with Jean Varney at the Mycological Association's 25th anniversary. And Ray and Jean made an 11-year study of the fungal diversity at Hutchison Memorial Forest. And a lot of those specimens are in the NJMA Herbarium. In 2005, through the help of Dr. Jean Varney, who I might add was instrumental in having me join the club, because I had picked up Dr. Orson Miller's book in 1971 or 72, after having moved from Brooklyn, and there were so many tropical storms that year. I lived near the Passaic River. There were so many mushrooms I tried to identify, and not sure of what I was doing, I called Rutgers to ask if there was someone who could look at my identifications, and they set up a meeting with Dr. Varney. And at the time, the club was had an article. There was an article about the club in the New York Times, I believe, and Dr. Varney showed it to me, and then, you know, that's when I joined. So he's the reason that I became a member. And but with his help, the Cook College Herbarium of Plant Pathology got moved to the Chrysler out of the attic in Martin Hall. And so our specimens also moved there as well. And if you are able to attend the open house on February 7th, you can read more about this Cook College, Douglas College herbarium. So it's 40,000 fungal collections, started in 1889. There's a lot of rusts and smuts, which are, plant pathogens, but we are lucky to be housed in that area. I cannot remember when the executive committee formally established the name of the herbarium as the Raymond M. Fatto Eugene H. Varney Herbarium. Maybe someone can look that up in an old newsletter. But the herbarium was given this name because for many, many years, Ray and Jean were the only ones entering specimens. Occasionally I would join, but they did the bulk of the specimens, particularly the ones for at the Hutchison forests, sorry. But there are many other important members of the Herbarium Committee. John Burkhart, who's the data manager, and Nina Burkhart, 4A chair of NJMA. They have been always involved since I became the chair. So here we see in this picture John, Jason Halfstad, Nina, and Lila at one of our days of entering specimens. So here we go. Nina and John, along with other club members, go monthly to Franklin Parker Preserve in Chatsworth. And this study is ongoing since 2009, which is quite remarkable. So they've really contributed a lot to our club, and many of our specimens are from the Franklin Parker Preserve. So some of the fun facts. In 2013 at Wells Mills, a blue mushroom was collected on a club foray. And here you see not the picture of that one that day, but others from Ann Beckler and Garrett Taylor. So Nina recognized it as unusual and dried it and sent it to Dr. Tim Baroni, who was a pink spored expert. Nina even did an article that's in the May-June 2014 issue of NJMA News, wanted Ellis's blue mushroom. Well, what happened was that Dr. Baroni identified it as Caloderma indigofera. And we actually located in the Mycological Herbarium from Cook Ellis's collection, and he called it Entoloma indigoferus from 1879. And so therefore, it had not been recorded in 135 years. You see a picture of Ellis's specimen, and notice it says, in deep swamps of Cupressus thyoides. Well, actually now it's Chemacypris thyoides, which is Atlantic white cedar. So this mushroom was added to the fundus study as part of our Northeast Rare Fungi Challenge. And so there's actually now over 200 images on iNaturalist as of 2025. And so it has possibly been found along coastal areas because that's where Atlantic white cedar grows. This is another connection. Another herbarium specimen from the Cook College specimens was collected by G.W. Martin in 1919. It's actually Sterium hirsutum. And this Martin was not the guy of Martin Hall at Rutgers, but he did attend Rutgers and he got his Master of Science degree in 1915. He then went to the University of Chicago for PhD and became a professor at the University of Iowa. And he co-authored, although I'm sure most of the work was done by Alexopoulos, on the myxomycetes, the slime molds. But getting that connection back to New Jersey, and there's the frontispiece of this incredible large tome, the Myxomycetes, again, Martin Alexopoulos. In the middle picture, you're going to see just a few of the beautiful microscopic sketches, drawings of made by Ruth McVeigh Allen. She died in 1984. She's from New Jersey, and Ruth illustrated over 360 of these specimens, which are all shown in the back of that incredible tome, the Myxomycetes. Very interesting, too, was that Ruth Allen was one of the original members of the Pine Barrens Coalition, and she campaigned actively for its preservation. So that is a really neat thing. She's also a member of the New Jersey Conservation Foundation. Supposedly, her own collection of slime molds was donated to the National Fungus Collection. I have yet to be able to find out if that has been correct. But this was the last slide at the presentation, and I wanted to thank all the club members who have given time at the Fado Varney Herbarium, and especially to Megan King, who's the associate curator, Dr. Lena Struver, who's in charge of the herbarium, and Dr. James White, our connection at Rutgers as well, specialist in endophytes. Here you see pictured Igor, Betty Wise, and Phil Layton. Unfortunately, we lost these wonderful devoted members, Nina. And over here on the right, Nina's on the floor. Behind her, Kira, Luke Smithson, Igor, and John Burkhart. I did add this just today. This was the Herbarium Open House of April 2025. And we see Donald, who comes from Brooklyn, Jason, John, myself, lady, I can't remember her name. This is Svetlana. Svetlana wrote an article in our summer newsletter of 2025 about her visit. Another lady I'm not sure of, Nina and Lila. And I added this because I knew I had a copy of this article by Susan Hopkins, which she wrote after a herbarium open house in the July-August 2005. Over 20 years ago, she wrote an article for our newsletter. So Susan checked out specimens of the tooth fungi, as well as hypogeous fungi, those the false truffles. So pictured at the bottom is Rhopalogaster transversarium, identified by Ray in 1991. That's a specimen from our herbarium. The previous November, Jim Trappy of Oregon came to speak to us. So I hope I'm going to exit. And am I back on or not? Oh, stop sharing. How's that work?
Great. That was wonderful, Dorothy. Thank you.
I'd be glad to answer any questions, but I hope people got a chance to look at some of the important members in the beginning of our club's history.
I love that, Dorothy. Thank you so much for sharing. And a couple of things strike me, just that that time in America, it seems like some other clubs started across the nation, like at similar time, and I just think it's interesting. I know our club in Illinois started like a few in the 80s, but it just it must have been fun back then to, you kind of felt like you were pioneering.
Very true.
Yeah.
The thing is, all the names I learned are almost all of them changed. So I have to relearn.
That's right. Yeah, but no, that was so fun. Thank you so much for sharing. And you mentioned Kibby is in Europe now, in London, did you say? Or was
Yes, Jeff
yeah,
Kibbe.
and
Yep.
he's still alive then.
Yes.
Oh,
Yes.
okay. Is he doing mushroom stuff still, or?
Indeed, he's even authored several books. He's an incredible artist. I have some of his drawings hanging in my living room and hall.
So he's still active in it.
I hope so. I really haven't kept in touch.
Okay. Yeah. Yeah, that was neat to hear, because I mean, I know the Fado and Kibby names and just kind of assumed they were like sort of long gone, and so that was a nice surprise. So yeah, thank you.
One of Jeff Kibbe's drawings on the artist conch, the Ganoderma applanatum, is in the office of the mycological curator at the New York State Museum. So
Oh wow.
I had the honor of seeing that. It was one he had drawn for one of the nymphs. So
Is it a picture of Peck?
yes, it's the picture of Peck. Yeah, who of course was the New York State mycologist.
Oh wow.
Peck. Yeah. So a nice sketch of Peck, and it sits on a shelf there.
That's
So
precious.
yeah. So yes, a lot of great history in there, and a lot of different people over the years. But certainly, Dorothy, you've had the longest continuous ongoing connection with the herbarium, 48 years of plus now of making accessions. So I think that's a remarkable contribution, and I hope
Thank you.
hope it continues for many more years.
Oh, yes. And we've got very interesting things in the works because one of the Rutgers students, she didn't receive all her grant, but a partial one. Her name is Kelly, and she wants to digitize some of our herbarium specimens. And hopefully we can support her grant with some of our NJMA funds.
Well, Dorothy, I had a couple of questions. One short one. So that blue Entoloma that was found in a cedar swamp, I think I heard you say a genus name for that I'm not familiar with. Um,
Caloderma indigofera.
Caloderma. Yeah, I don't know that. I'm going to write that down. Is that an actively used genus name? Do you know, or has that been maybe absorbed into?
I believe so. I mean, that's what Dr. Tim Baroni called it.
Oh, okay, and that was
if
just
you
a few
want
years
to,
ago. Caloderma. Okay.
and I do believe that's the name on the fundus rare specimens as well.
Oh, okay. Yeah, I'm not familiar with that genus name, so
Yeah.
I'll look into that.
I was
Also,
hoping Nina and John would have gotten on to this Taxonomy Tuesday, but maybe because of the new system, they weren't sure how to do that. But Nina wrote an article in 2014 about Wanted, Ellis's blue mushroom. And I have a write-up at one point too, and I think even Lila did. But interesting thing in that article, when Jim Richards was newsletter editor, he said, I can remember Bob Peabody repeatedly suggesting that NJMA organized some summer forays to look for the mushrooms that J.B. Ellis had collected. That was back in 1976. So Ellis lived in South Jersey, the Pine Barrens area. So, it was really cool that Nina located the actual dried specimen that Ellis had collected. Of course, he was calling it Entoloma indigofera at that time, or Agaricus, actually.
Mhm.
Yeah, all the guild mushrooms were agaricus for a number of years.
Yep.
Yep.
What-
So a couple of years ago, working with Jason Halfstead, I did a fungal survey of Haynesville Wood Preserve, which is in northern New Jersey. It's not far from Stoke State Forest. I have a bunch of specimens here that I dehydrated. I had hoped to maybe send samples into Ohio Mushroom DNA Lab, but they stopped taking submissions. But I still have a bunch of dried fruit bodies here, and I would have to go through them and see which ones look like they're still in good enough condition. But I'm wondering if these would be something that would be desirable to have put into the herbarium. I've got MO observation for every single one.
I know, according to Dr. Jean Varney, sometimes if it was something that was very unusual, even though it didn't have an identification, he felt that we should enter it. If, we've got all the other data about it. But so what are you saying, that these are not fully identified?
Well, some are and some aren't. I mean, there's a number of russulas, for instance, that I was only able to identify to genus. But there are other, so for instance, I have Inocybe subocratia, which Britt Bunyard put into Fungi magazine last year as this unusual species that's rarely found, but there were a whole lot of it out 2 years ago. I was finding it every place, and I found it at Haynesville. There were a lot of them there, so I have a collection of that here from
So
Haynesville.
we
Just as a
definitely
for instance. Mhm.
we definitely have things from out of the state. So that would
No,
be
this is New Jersey.
Oh, all right.
Yeah, this is in northern New Jersey. Haynesville is up near Stokes.
Well, if it's in good condition and you have all the information on one of our our tickets written down, and you have the MO number, sure.
Yeah, I guess I would need to have the tickets to fill out.
Right. Well, if you come to any of the New Jersey forays, there's always pads of them.
Those things. Oh, yeah, I think I might have some of those here. I'll look.
So
If not, I'll get some. That should be no problem.
Any other comments, questions?
yeah, I mean, how does a thing end up in the herbarium at this point? Like, what is today's version of that? Like, who decides what's interesting or whatever?
Well, I get to, some of the big weekend forays. I was at NAMA and NEMF last year, but I always do Meadow Woods because I'm nearby. I don't get to a lot of the forays down in South Jersey because it's a good 2-hour trip, just like some of you can't get up to the north ones. But so Nina and John, being our digitizer and foray leaders, they're the ones that keep these wonderful records and have them with them on our forays to see what's in the herbarium. And if something turns up on a foray and is pretty well identified or not, and is unusual, they dry it and bring it. bring it to the times we enter the specimens. Hope that answers your question.
Yeah.
It's Nina and John doing, this tremendous bulk of work.
But I think we're open to other people that have interesting collections.
Oh, absolutely.
Approaching the committee to have them put in, just like Dave did. I know also that Nina and John are looking for people to help so that they aren't the only ones taking interesting things away from the forays and drying them, but they want to spread that workload around starting this year in the club. So
yes, and certainly Igor too. And we have some of your specimens, Dave, from somewhere else as well. So,
yeah, so everybody really can contribute to this. But there's only, a couple of times a year that we meet to enter them in. So,
and I've dried specimens before and want to enter them, but I never put them on MO, and so now it's
yeah, they can go on INAT. We put them in. They're also acceptable with INAT numbers.
You know what, Dorothy, if you create INAT observations, you can easily import your INAT observations to MO 10 at a time.
All right. Well, hold on.
It's so easy. It's ridiculous.
Hold on. I'm not a techie person, so
neither am I.
No,
this is
let
very
me explain something. I have been doing INAT for a gazillion years because I not only do insects, I do all kinds of plants and so on INAT. Trouble is, originally when I had INAT on my home computer, I can't open that now to enter things. I only do INAT on my phone. And when you do it on your phone, you can't get that number. And it won't connect to my home computer where I'd be able to look up the INAT number.
Yeah, you can go and browse to it now, Dorothy. I could go and browse and get the number of any one of your INAT things now. Any of us can get the number of anybody else's INAT observation through our browsers on our computers.
Yeah. Dorothy, if you have an INAT account, you can sign in on your computer and just click on your observations, I think it's called, up top.
Yeah.
And then you see all of the things you've ever posted. Yeah. And then you can filter fungi or lichens and just see.
I know, which you can't filter on the phone.
No,
right.
No, this stuff needs to be done on a computer.
No, may I please add that actually, if you open any of your observations on the phone, it's very hard to see, but in the top right-hand corner of the photo on the phone is a share icon, and it's often lost in your photo. But if you click on that, then you can share it like with yourself. You'll get the number and can share it.
It also makes a difference if you're using the browser on your phone or if you're using the app. If you use the browser, you can get the number.
You can do it with the app as well. You just have to open your observation, and then you can't see the thing. But if you click I can't see it at all, but I know it's there. So I click the top right-hand corner of the photo, and then it allows me to copy the extension and then send it.
I've been using the classic. I have not been able to.
Me too.
Yeah.
Yeah, me too.
I need someone to show me. I'm very bad.
Well, you can't even see the thing. I mean, I needed someone to show me too because I couldn't see it. It's just not really visible.
I see it, Hunter. I would never have noticed that. Thank you.
Yeah, I just tried it, and yes, I can do it now.
It is just on the classic version, though. I have both downloaded on my phone, and it does not seem to be on the new version, just the classic.
Okay.
So, all right, I'm looking at something that I entered right now. Xanthomendoza weberi. It's a lichen. So
Just
where am I
click
supposed to look?
look click the right, like touch your finger on the top right hand corner of the actual photo.
There's
And
there's a little black circle.
yeah,
That's it.
click it. Yes. And then you should get like a copy prompt or something. Did anything happen?
I'm getting something, but I don't know what to do with it.
Well, if you copy it, then you can, if, I mean, I get copy as an option. And if I copy that, I could like paste it into an email and send it to whoever, or you can,
Text it to yourself.
or if you have a Mushroom Observer open, you could add those numbers that way, if that's the goal.
But I'm
Hunter, is that
not
how you
seeing
send
any
your
numbers.
email?
Dorothy, do this. Don't do it.
Sometimes.
Call somebody up and
Oh man,
silly.
that's snappy.
Yeah, I didn't know about that. I'd been opening the browser in my phone and squinting my eyes to see.
Hmm.
All right, we're gonna have a little session on how to do this. We'll have to figure this out. But Dorothy, I'll come down to your house and sit with you and we can play with any of this stuff anytime on your computer.
Okay, thank you. Because Bill just came in and is yelling at me for taking up your Taxonomy Tuesday time.
No, it's okay, Bill. It's fine. We're having fun and we're learning things here. So that's what it's all about.
Yeah, anytime you want to do history night, Dorothy, I'm here to hear that too.
I
Oh,
love
okay.
it. I
Thank
love
you.
it. Love it.
One other thing before we let you go. Last year we had a new club member show up. I think he's some sort of a researcher. I think he's a member from down. He had been at DC, or maybe he's still involved with the USDA or something, but he might live in Jersey now, South Jersey. And he wants to find a number of the Ellis species, if you will. So he was at Stokes or someplace talking about that. I can't remember his name off the top of my head, but it's interesting. All these years, everybody is still on these quests to find Ellis species in the state. So I think that's great.
Yeah.
Okay.
Yeah. That Entoloma was so nice to see too.
Yeah. All right. Are there any other questions for Dorothy about our herbarium? Just a reminder, a week from Saturday, we are having an open house there at 9:30 on the Rutgers campus, and that is followed at 11 by a guest lecturer coming in from Oregon, a gentleman by the name of Charles Bruder. So hopefully a number of you can make one or both of those events on Saturday, February 7th.
Oh, how important is an herbarium cabinet to preserve specimens?
The cabinet,
They're sealed very tightly.
yeah, better than a regular file cabinet. The
Right.
drawers are also designed to hold the collection packets efficiently, right? They're not deep drawers, they're shallow drawers, and you can,
Keep them organized.
yeah.
Yeah. But then it does sort of keep moisture out or something too, right? Yeah.
Mhm.
Cool.
All right. So now, yeah, it's almost 8 o'clock. Wow. All right, good. We will move on to looking at some member finds. If you have some and you're online here and you would like to share, speak up, or put your name in the chat. I'll give you a minute to do that, and I will go get ready to share from the computer. Anybody, anything they want to share locally? All right, not hearing. I'm going to pull up Hunter's. Hunter, would you like to go first? Sure, she wants to go first. Why not? All right. Okay, Hunter.
Okay, great. So this is probably my most recent find locally, and it's still probably a month old. Um, Dacryopinax elegans. And I've shown it actually before, not this specimen, but I'm pretty sure I've shown it before. I understand it's not that common in at least certain areas out east. And I mean, I can't say it's really that common here. Um, these are the spores, and
Super yellow.
I use Meltzers, and they have to me they looked like little butternut squash with a stem. Each one seems to have I think it's a piculate, or is that the right word?
Apiculus,
Apiculus.
maybe?
Apiculus.
Apiculus. Yeah. Um, and I think they're supposed to also be septate when mature, but I didn't use immersion oil. I wasn't able to capture that, nor did I look for the Y-shaped cystidia that they have. But I love this mushroom. I think it's beautiful. And I think it's also probably people walk right by it and think it's like Exidia or baby auricularia. Um, but it's kind of tomentose, I guess, at the base. And I like seeing it. I love seeing it. I love finding this one. And it's tiny. This is like smaller than a dime, very small.
What does the underside look like, the fertile surface?
Pretty similar. Pretty similar. You can see the top edge of that one in front of it kind of hanging over.
Yeah.
That's, yeah, I don't know. I should have looked at it like under the dissecting scope to really see. That would be a good practice for me. But at that size, it kind of looked the same.
Yeah, I was gonna say that thing's if you can section off a real thin slice, I bet those spores on the Bisidia are super interesting. They probably look crazy, like a set of bananas or something.
No,
Yeah.
there's one. I think there's one per stigmata on the.
Really?
So the basidia are Y-shaped, like the same as with Dacromyces. So these are related. I think they're in the same family. Um, so they're like these Y-shaped basidia, and I'm pretty sure that each terminal point on the bifurcated basidia, I think they have one spore on each one.
So
I
it's like
think
a
there's a bunch of,
bisporous kind of.
yeah, yeah. So it's like two spores per basidium. I'm pretty sure. I'm gonna have to look this up myself, but I think that's the way it works. So, and the basidia are really large also on these Dacryopinax and Dacromyces. mushrooms. I don't find these very often. But I've looked at Dacrymyces, and it looks to me like the spores are a little different than Dacrymyces spores because they're rounded on one end and tapered on the other, giving them that sort of a squash shape that Hunter mentioned. And so Dacrymyces spores tend to look like grubs, you know, like little larvae, I guess you would say. But
More even though.
yeah,
Yeah.
they're more symmetric in terms of the two ends look the same, but they're usually also curved a little bit as well, and sometimes septate. Um, so what was this at 400 power? This
Yeah.
picture? Mhm.
Yeah.
Um,
Yeah.
yes, I think that would be one difference possibly is the way the spores are shaped.
Just
Dacryopinax and Dacrymyces.
And I do want to correct myself. It's the Y-shaped Basidia, not Cystidia, I said.
Oh, I'm sorry. That got past me.
Oh, yeah.
Yeah,
Well,
yeah, it's Basidia.
yeah.
Right.
Oh, you
Yeah.
got it right, but I got it wrong. Yeah. Got it.
the old timers used to say that it was the shape of a tuning fork.
Oh, right. The Basidia.
Yes.
Mm-hmm. Yeah, I like that.
That's still in some of the field guides and even on the online descriptions. You'll see the tuning fork. Tuning fork Basidia, they call them. Mhm.
Great.
That picture of the fruit bodies is really nice.
Yeah, that turned out nicely. I did receive a lens that I think is going to be pretty good. I haven't used it yet, but for Christmas for my phone. So hopefully things will start looking up photo wise. But yeah, this one turned out nice.
Yeah, how did you manage to get? I mean, the one that's in the upper right is sort of in the backdrop a little bit, and it's still pretty clear. It's only a little bit,
A little
um,
scary, but
just a
yeah.
little bit, but that just helps give it a little nice depth of field, also. But it's not real foggy, like it shouldn't be there, you know.
Yeah,
Because these
very
are so
nice.
tiny and so close together, maybe.
exactly.
Oh,
That's
okay.
all I can say. Exactly. That's exactly what I was going to say. I have nothing else to say, but yeah. Yeah. Yeah, this was a good one.
All right. Very nice. Next one up.
Oh, so I've already also showed this fungus before, Dendrothele nivosa. But this is a better specimen. This is actually from just over the border in Iowa, and it's a crust fungi that grows on juniper and I guess red cedar. And I have a picture of like the whole tree too. But I just saw it again, and um, now that you see it, you'll start seeing it everywhere if you have the juniper. Um, and I did find out who had been studying this um, at the University of Wisconsin, and unfortunately, he's become an English teacher in Mississippi. And but they may still publish the paper at the Anne Pringle lab on this. We shall see, or on this genus. I guess there's a few others in the genus, or maybe quite a few others. So I'd love to see some more if anyone knows any others.
Yeah, I don't know anything else, I don't think, in this genus.
Okay.
This reminds me a little bit of the Xylobulus frustulatus.
Mm-hmm.
Yeah, it's interesting.
Yeah, the way it's breaking up a bit.
Yeah, a little bit.
Yeah, these were better than the ones I'd seen before, but I'm sure this is what it is, don't you think? I think. Just because it's on juniper, living juniper. Yeah, and apparently it doesn't really kill the trees or compromise them much at all, but it is a parasitic fungus.
Mhm. Yeah, I think you showed it last summer maybe, and I think then I did spot it on a
Oh,
juniper
cool.
somewhere.
Oh,
Yeah, I'll have
good.
to look through my collections and see
Yeah,
that. But yeah, because I remember you talking about it before, so I did start to look. I don't run into juniper that often, but
same.
yeah.
Right. Yeah.
Okay. All right, good.
Oh, yeah. So this is a really old picture that has been labeled Inocybe. I got some pretty reliable feedback that it is a Inocybe. But I just kind of wanted to relook at it. It does have those kind of striations on the stipe that would be a Inocybe like. Um, and but yeah, I'd like to learn more about this genus, and if anyone is able to put a species name on it, or talk more about Anosibes. I do think they often have that hump on in the middle. It's like more than an umbo in this case. Um, but anything anyone else wants to add about Anosibe or this Anosibe, or if they don't agree, these were found in basically a path in a oak woodland, oak kind of bottomlands with cottonwood and maybe some hickory, but definitely large oaks and cottonwoods, but right on the path.
So I don't know what species name to put on this, um, but it's pretty clearly, I think, something in one of the genera in the Inocybaceae. So it's only a few years ago that Brandon Metheny reorganized what used to be genus Inocybe into now what is being called family Inocybaceae, and there are, I think, 4 or 5 genera that are commonly encountered in eastern North America, and Inocybe is still the largest of those genera. Still has the most species in it. But there's also Pseudosperma, Inospirma, Malocibe, and I think there might be one more, but perhaps not in eastern North America. So it's those 4. So the first thing you want to do, if you're pretty sure you have something in the Inocybaceae, is try to get some spores. I always try to get a spore print from a drop from even just a little piece of the cap, but also scope the gill. So it has to be not that difficult to scope because the cystidia on the gills are fairly prominent. And there's 2 types. There's chylocystidia that are on the gill edge and pleurocystidia that are on the face of the gill. And genus Inocybe, those are the only ones that have pleurocystidia. And the pleurocystidia are always very distinctive, usually with apical encrustation or other sorts of ornamentation on the apex. And they're thick walled. So you can see they have really thick walls, just the way the light is being emanated from your microscope goes through the wall. And you can see it's thicker, toward the margin. Um, but the other genera, Pseudosperma, Inosperma, Malocibe, almost always, if not always, I'm not sure if this is an always thing or an almost always thing, will have a lot of chylocystidia, and they'll be in great big bunches. So it's what's sometimes called a palisade, just this row of cystidia that just line the edge of the gill. And those are not as distinctive as the pleurocystidia that you'll find on a true Inocybe. The Chylocystidia on fruit bodies of the other three genera are they look like bundles of balloons, really. They're thin walled. They're usually just sort of clavate, like a club shape, or maybe um, a little bit inflated at the apex, so it's a subcapitate. Um, but that's to get started. If you can find the pleurocystidia, and they'll be all over the place, you just smash mount some gill material, and if you find the pleurocystidia, you'll see them all through the mound, here and there. So, and the chylocystidia, even if you just do a smash mount, you can usually identify where the edge of the gill is, which is sort of doing things backwards, which is because you're supposed to know where the edge is to begin with. But you can usually find where the edge is by just finding where all the chylocystidia are. And if it's one of the genera other than Inocybe, you won't see the pleurocystidia. There won't be any there. So that's the main difference used to segregate genus Inocybe from the other things that used to be in genus Inocybe, the Malocybe, Pseudosperma, Inosperma. Those will not have pleurocystidia, but they generally have a lot of chylocystidia. Also, if you find spores that are nodulose or stellate, or kind of irregular, almost like Entoloma spores, then you more than likely have a true Inocybe. However, the converse does not hold. Some true Inocybes have ellipsoid spores. So it's really a lot of microscopy to get to know the family Inocybaceae.
And for the moment, all of those in the genus Inocybe are to be considered deadly poisonous, correct?
They're not. I don't think they're quite as poisonous as the Amanitas in section Phalloideae or like Galerana marginata, but they're very bad. They'll definitely put you in the emergency room, is my understanding. And I don't know if it's true of all of them. And the family Inocybaceae is very large. You've got these 4 genera typically encountered in eastern North America, and there's a whole lot of different species. And I think there's probably still finding some new ones. So are they all deadly? Are they all very, very poisonous? I think that should be assumed.
Could we please see some of the other pictures again, especially the caps? Yeah, I might have a name for you. As little as I know about Inocybe, almost nothing. I can actually give a name right now for this one.
Wow, great. What do you got?
I think this is Inosperma vinaceobrunneum.
You want to put that in the chat for us, Igor? Thank you.
Yep.
Just for the record, it was unpublished, but Ray Fatto also did a multi-access key to Inocybe. Of course,
wow.
this was well before Matheny's work with the family Inocybaceae, but this work is excellent, and it was very helpful. Of course, you do need the microscope, but it was very helpful in keying down some of the names, which, if you did come to a name on his key, you would be able to then look up what it should be in the Matheny scheme. So if anybody is interested in that, I could send some copies.
Yeah.
Yeah, I'd like to see that.
Yeah,
Yeah,
that's
because those old names you can just cross-reference. A lot of times on Index Fungorum, you can just get some information about current names.
yeah, that'd be cool.
And a lot of the species names are more or less the same, even if the genus changed. Now, of course, there's the usual thing with there are old names that were European that are no longer apply in North America, and that's always a problem because you're still going to see the old name on Index Fungorum, and it's not going to be synonymized with a newer American name because they are not the same species. So that's a little bit of a problem trying to cross reference. But yeah, if you get the latest Methane Inocybe.org, because he keeps updating that. I'm not even sure I have the latest one. Then a lot of times you can get some information on your Inocybe. Although on Methane's website in his key, which he's calling a key, but it's really just very, very brief descriptions of a number of different species, North American species.
So, Hunter, if there's some way I could get your address or,
Sure, I can put my email in the chat. Should I do that? Or is it paper?
it's paper.
Oh, okay. That's great.
I have it. I'll get it to you, Dorothy.
Okay, thank
Lila.
you.
Yep. Yeah.
Hey, papers. That's nice. I like it.
I live with paper.
Yeah, I like paper, honestly, big time. So that's good. It won't get lost. Yeah. Good. Thank you, Dorothy. That's so nice.
Okay. What do we have here?
That the spores, pink spores?
Looks like it.
Yeah.
Yeah, one dropping.
So are these concentric sort of circles on the cap?
Well, okay, I was just
Or
talking.
textures?
I was muted, but it sort of looks that way. And I know INAT was throwing out lactarius, but I don't really think that's what it is.
Well,
No,
if it's pink spores, it's not lactarius.
Yeah.
Yeah.
So there's this thing that's been pushed around in all these different genera. Clitosella, Rhodo, something or other. Now there's another name that I've just been seeing. But they're in the Entoloma. They're
Yes.
pink spored. They're not the spores are not particularly angular, but I think they are a little bit. But I think that's probably what this is. It's one of those.
Okay.
Yeah,
I was thinking Entoloma as well. But yeah, INAT didn't throw any of those out at all. So okay.
Yeah, but it looks like that looked like a spore deposit on that one cap. There was probably another cap above it.
Yeah.
Yeah. Look, you can see the lines from the gills, sort of. That really looks like a spore deposit.
Yeah, so you think they dropped that way? Yeah. Yeah.
There was another cap above that one, probably.
Yeah. Yeah, that's cool. Yeah. And that's the right color. Yeah.
That's my guess. Let me see if I can come up with a name for you here. I'll come.
Igor, were you?
I for some reason I thought they were more colubioid, reminiscent somewhat of Lepista. But
yeah, that's a possibility also. They look more like these other things to me, but
Yeah,
that's
I
a
don't know about these other
possibility.
things. I never heard that name before. So
Yeah, let me see if I can find some.
Did you say Lupista, Igor?
oh, well, now it's in Calibia, but that's being sort of.
Oh, right. Hmm.
Do they have such crowded gills? Are there some with these are super crowded?
Yeah, that's why I don't think these are Entolomataceae.
Mhm.
Yeah, these look like a thing that I found last year that we kind of came up with the Calibia name for,
But those,
because they grow in those clumps like that,
Yeah, Calibioid. Yeah, like
and they have that rolled gill edge.
I
Like,
also would not be surprised if these had a pleasant odor.
yeah, and I do not know. But those would have more a white spore print, no? Or
mine had a pinkish.
oh, okay.
Where was this collected? Mississippi?
I think this is from Illinois.
Oh,
Um,
Illinois.
I'm not sure if we,
Yeah.
it's another like,
I see. This is from August 24. Oh, I see. Okay.
yeah, but it's a big guess on where it's from though. So
August,
that's why.
August 24.
Oh, you don't think it's necessarily from Winnebago County?
No,
Oh,
that's why it's like a huge circle. Cause yeah, no, yeah, but probably I think it's probably from Northern Illinois.
okay. It looks.
And what about, something like a Clitopolis prunulus, which is pink spored?
These are
And
too
how's
large.
that? What?
I think these are too large for that.
Too large, but
Yeah.
looks like a
I thought of that. They reminded me of that too
Oh,
initially.
But
I,
the
I'm
gills
going
should be current,
I'm
I
going to
think.
put something into the chat. Also, Dorothy, you had mentioned Rod website being semi-accessible these days. The website's been updated, and somewhere in the chat I put before. If you
yeah,
scroll
I
through
saw
chat,
that. Great.
yeah,
Thank you.
that's the new version. It's been working really well for me. Although it's not, it's sort of funny because I think it's an older version of his website before some of the provisional species names were added, that his son probably just used as the prototype to, you know, um, to make the new website. Because it seems to be missing some of the newer provisional names. But anyway,
Yes,
I'm gonna put something into the chat now. So Lulesia is this new genus name for what used to be Clitosella, and then it was Rhodo something or other. I forget, but I just put into the chat, um, what my guess would be, for this thing. As Igor pointed out, though, the gills are a little closer on yours. But you can see here, see how like the
Similar.
caps are like kind of concentric. Now this is going to be pink spored. I'm pretty sure these are pink spored. You can read on the Mycological description. So the gills are pretty close, slightly decurrent, not strongly decurrent, slightly decurrent. They have the caps tend to crack up on these, as you saw in the one picture, but not always when they're younger. The cracks don't form until a little later. That would actually be my guess for that thing that you have.
Okay, thanks.
Something along
Yeah.
these lines.
I don't think it's this.
I don't think it's this either.
Where is her thing? Yeah, these look notched. These gills.
Yeah, they're not coming down the stipe
Yeah, they're
No.
not
same
such
way.
so decurrent, and they're very, very tight.
Yeah, there's a few other species though in this genus Lulesia. I just
Okay.
pasted the one in. Let me go back to Michael Quebec here. Where did I have this?
Oh, this would have been a good specimen for sequencing.
Yeah, that's a good, whatever it is. That's a pretty good one. Let me see.
I mean, I definitely see similarities for sure.
Yeah, I think Mycological has two other species under this genus Lulesia, which is a fairly new name to me. These guys used to be in something else.
Or these look
Um,
closer to Quatapilus than hunters, but what do I know?
yeah,
Well,
they're similar to Cletopilus. Yep, they are. Well, let me see. There's also this thing, Lulesia obscura. What does that look like? Um, I'm just looking on a different window right now.
Very
Oh, those are
pinkish.
yeah, those are way different. Okay, so that species is way different. Um, I'm pretty sure that pink spores, though.
okay. Well, some ideas to look.
Yeah, some ideas
Yeah,
to go on.
yeah.
All right.
Lulesia, Colorado. What's that one? I don't know that one. These names are new, probably.
Probably.
Yeah, this one's kind of gray. This Lulesia, Colorado. That one's more gray. That one almost looks like the aborted Entoloma mushroom.
This is an Amanita persicina. I don't think I showed this one from Mississippi, but if I did already, I apologize. It's very pretty. This is in the Amanita muscaria group. And so it contains the same toxin that actually can be cooked out if done properly. But this was just a really beautiful specimen, I thought, and so I threw it in.
Gorgeous.
Yeah, really pretty. I love um, the stem, how it's got that orange on it, and the veil remnants.
This looks like it's where you touched it when you
It seems like it, yeah.
yeah, otherwise it's sort of flocose or something. Yeah.
Hmm.
And Igor, what's the term for this type of volval ring here?
I'm sorry, I was momentarily away from my computer.
Yeah. How would you describe the base on this? Is that rolled sock or what do you
No,
call
these are just the typical concentric rings
Okay.
of the muscaroid types.
Yeah.
Yeah.
Yeah, these are muscaroid mushrooms. They're not pantherinoid.
Yeah.
So
Right. Okay.
yeah,
Yeah.
Yeah.
The pantheroid has the rolled,
yeah,
but
just
beautiful.
refer to that paper by Gemmell and Tulas from 2008. I think I said that all the species that they sort of propose there, I mean, or entities, they say they're going to be all different species. It's an interesting paper, but it's quite old, 17 years. So, but this thing there is there too, and it's not named persicina yet because that name um, uh, that was a variety, I think, of Muscaria. It was promoted to a species in 2015, I think, also by Gemmell and Tulas. So yeah,
Yeah. Okay. Well, thanks for letting me share.
yeah. I don't know if they find these in Cape Cod, but we do find this as far north as maybe about New York City in New Jersey, the same latitude. Maybe with the global warming, this thing is going to be more widespread up north, in some time.
Pretty one for sure. Nice photos,
They're
Claire.
really pretty.
Yeah, I wouldn't call it common, but I know of a couple of spots where it grows almost every year. Um, One of those sites is FPP, I think. Yeah, FPP at Speedwell.
Mhm. Yeah, he found it in December down in Louisiana, it looks like. Yeah. Nice.
Yeah, their winters are full of amanitas, which is really nice.
So Hunter, is Logan still active in mushrooming and
Yeah, yeah,
Turkey?
yeah. He's not the president anymore. But he was at the annual foray that I went to, and yes, still very much so. Yeah, Reese Krantz is the new president, and he's so sweet. You would really like him. He's really nice.
Yeah, I never met him. I don't even know the name. The reason I asked about Logan is that he's been kind of absent from MO for a while, so maybe I thought he moved on, in the sense that he kind
You
of gave up mycology or something.
you can find him on INAT.
Oh, that's where everybody is. I see.
Yeah, and also GSMS Facebook page. Yeah, he's still around. Yeah.
Yeah, there's even less likelihood that I'll ever be on Facebook, but more chances on INAT. But I'm still holding off.
Understood.
All right, Dave's got a Lentinellus here, looks like.
Yeah, so these are probably all Lentinellus ursinus. But if you read the mushroom expert take on these Lentinellus, these bare Lentinellus, there's like there have been other species names proposed. They tend to name them after animals. So Lentinellus ursinus is named for bears, and that's probably what this is. I did find another collection on the same day that weren't not as darkly colored on the cap surfaces, and the spores seem to be maybe a little bit smaller. So this was during Mycoblitz. This was a few weeks ago when when it was winter, but not really winter like it's really winter now. But so we had that thaw for about 9 days, and that coincided with Mycoblitz. So I asked people in the club here if they wanted to have a foray right in Wilkes-Barre. You know, people, a lot of our members live right in the Wilkes-Barre area, so people turned up. There were like 10 people, and we found some mushrooms. And so this is one of the things we found. I sent it into Mycoblitz, a sample.
Very cool. I really like this mushroom. I like I like it. I found it a couple times. I really think it's cool.
It's probably the most disgusting tasting mushroom you will ever find anyplace.
Yeah.
It is just absolutely horrendously tasting. I tasted it once a long time ago, and I have not forgotten that experience.
I must say it doesn't look very appetizing.
No, no, it doesn't. When they're younger, they're not quite as ruddy looking, and then they could be confused for oyster mushrooms, I guess, by a novice. Even when they're young, the gill edges are fairly sawtoothed. You can't really see it on this picture. I should have sent the other observation in. That one you can really see the sawtooth edges on the gills. But these they always have that. You always find some gills with sawtooth edges on these. So that was it. So that was what, January 11th, I think. We had this foray. And then there was another thing that I found. I found this next thing. Wait, is it? Oh, wait. Oh, this thing. Oh, somebody else found this. Actually, I don't know what this is. It's growing on a big piece of wood, like a big downed log. Um, I honestly don't know what this is.
That's
I didn't.
interesting.
I didn't try to do any analysis on it. I just I dried it out, knocked it into two pieces, and sent half of it to Steve Russell for Mycoblitz, and I kept half. And rather than just try to hack it apart to do some microscopy, I just figured I would save it. It's a small fruit body. It's pretty interesting though. I don't know what it is. Growing right on a big piece of wood in
Ugh.
January.
Dorothy, do you have any ideas?
Not yet.
Okay.
It looks a little branched.
Yeah. It's branched. It's dark at the base.
I suppose, you might think Radiolomyces, but Radiolomyces usually has it grows from like what do they call it? Exo something. It's like a white coating on the substrate. And
Right.
yeah. So this I don't see this. I don't see that trait for this. Yeah, and some of the ends of the branches, some of them are branched right at the tip. I don't know how long this thing was there. It looks to be in fairly good condition for, mid-January. I mean, there was very cold weather before the thaw.
There's
We had this 9-day thaw, but there was very cold weather before that. It was in the single digits several nights.
yeah. There's no way it could be like Artomyces trying to take a notion.
No, it's not. It's not Artomyces. It's definitely not that.
I'm remembering something like that we saw at Thompson Park the few times I've been there, and someone was thinking that it was another species of the Radiolomyces, but then
Yeah.
that got turned around and it was said no. But so I'm not sure what it is, but
that other species of Radiolomyces got merged into Radiolomyces copelandii, I'm pretty sure.
right.
Yeah.
Is that stiff or squishy?
It's not squishy. It's not real stiff either. I honestly didn't play around with it a whole lot because there wasn't much of it, and I just wanted to preserve it.
Okay. But you sent it in?
I sent in half of this fruit body to
Yeah.
MycoBlitz. Yeah, so
Super.
there it is. There's your field data slip for MycoBlitz. And apparently the mail just went out today because the snowstorm held things up.
Okay.
So, I mean, maybe in a year or so we'll find something out because
All right.
you know how it is with Mycota. He's always so backed up with things to do. But that's an interesting one, isn't it? Yeah.
It is. It is. Yeah.
Yeah, really neat.
All right. Cool.
So the next one was also found. The third one there that was also found during this little foray that we had. This is a little pleuroid thing. And honestly, I've got to say, I thought probably the best chance for a genus was probably Hohenbuehelia. And that turns out to be what it is. And I think it's Hohenbuehelia griseum. There's also another name, Hohenbuehelia, something longer. I think it might start with like cyano because sometimes it's bluish looking. But anyway, one of the people on the foray, one of our members, posts everything on INAT also, and he just lets INAT say what they think it is, and they thought it was Crepidotus, so they were wrong. So how do I know this? I didn't know what it was when I found it. I just wrote down Agaricales. Here's the key: these cystidium. Look at this cystidium, and I didn't find a whole lot of these cystidia. Because this fruit body was probably old. I'm guessing it was probably there for a while and probably froze and thawed. And so, but I did find some of these cystidium. So look on the lower left part of the cystidium. It's like this like incrustation of pointy stuff, just covering the end. And then on the other end, it's like this little handle sort of. And you can see the thick walls also. Do you see towards the outer parts of the midsection? Do you see how the wall looks thicker? So this is a metuloid. You know, it's a thick-walled cystidium, pleural cystidium, I imagine, because I think that's the only time you're going to see ones that are this kind of crazy looking are pleural cystidium. And I found a few others as well. Free floating. Took a while too. I had to do a little work on these to find some of these cystidia. But if you look on, I think it might be Mushroom Expert, or oh no, it's Mycological. They have a few of these oddballs, Hohenbuehelia species. They have a page for one or two of them, and they have these weird cystidia. This is the giveaway, really, is these cystidia with this weird ornamentation on the one end, and then the other end is just kind of tapered to like a handle sort of thing. So this is a Hohenbuehelia. And these cystidia are not in the best shape, probably because the fruit bodies themselves have been through freezing and thawing. So, but you can see here, I mean, the shape of these cystidia from photo to photo is pretty consistent.
Oh, yeah.
So that was a pretty good find, I thought. And
what are you calling it?
Hohenbuehelia gricium, maybe. I think I just have it as Hohenbuehelia on
I
INET.
see.
Yeah.
Okay.
So originally I just dried them out. And then, you know, the guy in our club, his name is Rick, he put it on INET. INET said it was Crepidotus variabilis or something. And I was like, geez, I don't know. That's not a Crepidotus. So then I felt like I had to justify, you know. So I took one of these tiny fruit bodies that was dried out, and I really didn't want to mess around with it. I did not want to mess around with these when they were fresh because the material was old and soft. And if I started hacking away with a razor blade, I was probably going to ruin the collection. And I wanted to be able to send in a sample for MycoBlitz. And you can see here how much that's how much there was once it was dried out. So I actually, after as an afterthought, I thought, okay, fine. I'm going to try to do a little microscopy on the dried material. And actually, I don't like working with dried material that much, but this time it was very useful because the fruit bodies were no longer soft and squishy and easy to just destroy if I tried to do anything with them. I was able to cleave off a piece of gill pretty easily on the dried material. And then I just had to let it soak for a while. And I think I started out with 80% rubbing alcohol and then moved on to like H2O and then finally put some Congo red. That's why they look red in these photos, because those are stained with Congo red. And I did find cystidia. I didn't find them embedded in the gill material. They were free floating, but you know, it's not just a piece of debris or something, because these are pretty consistently shaped. And even this one they're looking at right here, I think it might be Mushroom Expert has a photo of a Hohenbuehelia cystidium that's as crazy looking as that one. Like it's got this big hat on or something. So yeah, so I'm convinced, but it's sent in. I think it's probably going to come back as Hohenbuehelia gricium is going to be my guess.
Cool. And yeah. Okay, never mind. Yeah. Oh, yeah, I was gonna say in the one on the left, it looks like it has a really legitimate stipe there, or am I just
Not real. Maybe very short, like a millimeter long or something. Maybe 2 millimeters long.
okay? So that's
Barely,
sort of an
barely.
illusion on the left, that the kind of red where on the one on the left. Yeah, that the stipe?
No, that's
Oh,
not. That's just some wood, I
okay.
think. These were on
Okay.
wood. I
Okay.
think that's just some wood.
Thank you.
Sure.
Interesting. All right. That's good, Dave.
Yeah, it was good. We had a foray in, mid-January and found things. That was pretty good, I thought.
Yeah. Ooh.
Well, this is old. Okay, so I've had these things sequenced. They grew in my yard for 5 straight years. Last year was the first year I didn't see them. This is one of the earlier ones, the second year. So I had these sequenced ITS. So it's a morel in Morchella section distantes. It's the black morels. You might say, well, black morel? That doesn't look black. Well, black morels, even the common black morel, Morchella angusticeps, starts out being very pale, pinkish, beige colored, sort of, until the ridges darken and it becomes a truly black morel. So anyway, so I had these sequenced, and there were two names that seemed to be competing with one another. One of them was Eohespera, but that was an old name that's been reclassified as Norvengiensis. And the other name that was competing was laurentiana. So you'll see now that on INAT and on my mushroom observer observations, I've got a bunch of observations for these because they came out 5 years in a row. Some of them I just called Morchella section distantes, but now I'm calling them laurentiana, the ones that I'm putting a species name on. So you might wonder why. What caused me to change my mind? Somebody came on INAT, on my INAT observation of one of these, and ended up looking at some of the sequences that I had posted on Mushroom Observer, and pointed out that Norvengiensis and laurentiana are very, very similar in ITS, but there are two positions in the ITS where the base pairs will give you good evidence for laurentiana versus Norvengiensis. Now, I honestly don't know, myself about this piece of information, but this person produced a nicely written comment on my INAT post and explained about it being just these two base pairs that really signal a difference between laurentiana and Norvengiensis. So I'm calling them laurentiana now. And if you do a close up on this spore, if it's possible to do that, that's pretty cool to see.
Really?
Sometimes these Morchella spores have these little like gutules, but not inside the spores. They just kind of stick to the perimeter. And this one is showing that. So this is mounted in cotton blue.
Looks like a gemstone surrounded by little diamonds or something.
Yeah,
It's
like
bright.
little diamonds or something. Yeah,
Yeah.
yeah. So, but this is actually a trait that you will see in written descriptions of some Morchella species. So
Yeah,
there's
are outside, not inside. Yeah.
Yeah.
yeah, they're outside. Yeah, so it's probably just who knows where they're coming from. I don't know where those little bubbly things are coming from. So those are just the assayi. I mean, you
Wow.
don't get a whole lot of information from the assayi, but for some of these section distantes species, you do get some information apparently from scraping inside one of the pits or scraping from the ridge. And there are structures that supposedly will help you identify the species. But quite frankly, I did that, and I looked at the micro, and I came to the conclusion of Norvengiensis. So I don't know. The book is still open, in my opinion, as to what this might be. And I'm going to look through my collections and see. I may have saved a few more fruit bodies of these. Oh, look at this gigantic spore. What's going on there? Most of these spores were maxing out at around 28 microns long. But how long is this one? I didn't measure it, but it looks like it's probably 40 microns long. but that's another trait that I think comes up sometimes in discussion of Morchella, that there will be some super spores or whatever. I'm not even sure if that's a spore, honestly.
Did you look at the distribution for this name that you're using now?
Laurentiana.
Yeah, I'm just curious.
It's it comes up in the blast on GenBank. I don't know
Oh,
really where to find information on descriptions of these. I
it's
think I
probably
did.
not even.
It might be on Mycological. I think the Laurentiana might be on Mycological.
I
I'm
see.
not sure.
It's not, yeah, out there enough yet. Okay.
Yeah, these are new species from only like within 10 years.
Right.
Laurentiana, norvengiensis. Now, here's the other thing, too. I had always thought, since these started coming up, that they were the result of my having planted a morel grow kit in this spot under an old apple tree on my property. So this person who also seems to know something about this and pointed out this aspect in two positions in the ITS sequence, said that he doesn't think it's from a grow kit because this species was only described 10 years ago and the grow kit was purchased in like 2005 or something like that. But that, you know what, that's not really a good piece of evidence, because some company may have assembled grow kits based on a species that they didn't really know what it was, or they thought it was something else.
Right.
When I Google it, I get like all about cultivars. Like you can buy a syringe with this in it for growing, and
Oh, that species name?
yes,
Oh.
cultivars and
Oh, well, there you go.
yeah, that's interesting.
So this is one that is marketed as um as a grow on, on your
Yeah.
land kind of thing. But this was a long time ago. I think I bought this from someplace out west, out Oregon. I don't think it wasn't Fungi Perfecti. I looked and looked and looked for information on but on the purchase. But you know what? I don't think I bought it. I think my brother bought this grow kit for me and gave it to me as like a Christmas present. And because I had purchased a grow kit right after my wife and I moved in here, and I got nothing out of that grow kit, so I just gave up on that idea. But then he bought one for me, and that was probably this one. So here's two versions. I guess one of these versions of the ITS is edited. The one that's on the left, that's the one I believe that's the one that's in GenBank. So this was done by Fundus, I think. I'm pretty sure. This sequencing. So this was like it's probably a Sanger sequence, and it was edited and put in GenBank. And over on the right, I guess, is just the longer version. It's not edited. But I just pasted both of them right into this observation. So if anybody wants, they can see them.
Cool.
So the last time they came up was 2024, and there were like 9 of them. There was another year where there were 2 of them. It might have been 2023. There were 2 of them. Before that, the other 3 years, there was 1 each year. And so the fact that there were 9 of them in 2024, that kind of makes me think that's not a real good sign.
Yeah.
Because that's a Morchella trait, is to, put everything it has when it knows that it's not going to have much more time. It just puts
Run out of
everything
its
it
food
has,
source. Yeah.
into reproducing. And I think that might be what happened. So I'm not real hopeful I'm going to see any more of these, but you never know. We'll see. And then the last thing here is another thing from a few years ago that I sent. I'm not even sure where I sent these. Maybe Steve Russell. I didn't know what the heck they were. There's probably an MO. Oh, I think I identified these as some I call foliota or something.
Phaeomarasmius.
Phaeomarasmius, but it's a weird species name too. It's like it's the first time I ever saw this name. Phaeomarasmius proximans. So when was this collected? 2019, I think.
Yeah, late
You know,
August.
I think I probably sent this to Steve when he asked me to send him. I should have sent him like 500 things, but I sent him. I didn't realize he wanted like unlimited amount of stuff. I would have sent like 500 things. I sent him like 120, 150 samples. This was one of them, I guess. And it's only recently have I got this information back on it. But that was a pretty cool thing to find out because it's now there's yet another weird species that's not all that often reported that I know I have found on my 4-acre property. So it's a new one for me. I don't know if I probably tried to do some micro on. Yeah, I did. Okay. At least I got some spores. Yeah, it took me a while to figure out, honestly, that you really want to look around on your spore mount and find something that's not as messy as that particular photo. Then I also tried to take photos through my binocular scope. They don't come out very good. But what I should have done was, and I know a little better now, I should have taken like that first, what you saw in the first photo, and I should have maybe taken it off the microscope, maybe tapped on the slip a little bit more, got the spores to spread out a little more, and then put it under the scope again, and then looked around for an area where there weren't too many spores. There's too many spores in this photo. It's just messy. They're getting in the way of each other.
It's very hunter-esque.
Oh,
I've seen that before.
right. Yeah. And this is just something I learned from experience. You want to, in fact, somebody on MO a long time ago told me, try to find a view where you see like 40 spores. And that got me thinking like, oh, I guess it's not a good idea to try to get information from a situation like this. But you live and learn and revise your methods. And then I guess I tried to get a photo of a gill. I don't see much going on there really.
There's some finger here.
Oh, there's some, okay, and who knows what that might be.
Mm.
I don't know. It's not the greatest microscopy. I'm not the greatest microscopist, but I've got a little bit better over the years. So anyway, it's a species name I had never heard before, and it showed up one day. I got up and looked on my computer, and so I get these notifications in my email. Such and such MO observation is such and such name has been proposed. And I thought, what the heck I never saw that name before, and I looked, and it was this. So.
So what is it again? Sorry.
Phaeomarasmius proximans.
Okay. And that was on wood?
Does it say anything in the notes?
Just looks
Analog.
Came off wood, but I'm
Analog.
You know what? Click on the link to Mushroom Observer because I might have put more information there. Because this was back when I only posted on Mushroom Observer. And on a log. Yeah, so it was on wood. It's on a log.
cool. I feel like I might have seen that before, maybe.
Yeah, I kind of feel like I've seen that too.
Do I have a proposal here before I found out what it was?
I think I have to log into
Oh, yeah. Yeah. Okay.
Let's see. Foliota
Foliota
granulosa.
granulosa. Yeah. Okay. Yeah.
Yeah, and Mycota came on and said proximans.
No, I don't think, did he put a sequence up? Sometimes he won't put a sequence up if the sequence is not particularly good. But if he thinks he sees enough information to add support to what he believes the thing is based on morphology, he'll come on and give a high confidence name. And I will usually trust him on that, even if he doesn't post a sequence, because the sequence might not be all that good. Um, but it used in conjunction with, this, for instance, the sequence might be good enough to say, okay, it's not Foliota, so, um, I just kind of trust him on that. I didn't vote on it myself. I'm just letting the ID stand as his proposal.
Yeah, one of the things that's missing here is you don't know when Mycota came on and made that proposal.
It was not long ago.
Yeah.
I think, yeah, it would be nice if they dated when the proposals were made. Yeah,
Yeah.
they don't do that. Yeah, yeah.
It's been viewed quite a bit, 116 times, so
Oh,
yeah. Okay. All right. Was that it? Was that the last
yeah,
one?
that's it. I just thought I figured probably that would be bad to have some stuff to talk about, because I imagine nobody's going to find anything now for a while. Is there a lot of snow? Is there snow in southern New Jersey? Or did you just get ice down there?
Yeah, we got snow too.
Okay. Yeah, we got 11 inches of really pretty dense snow. It started out as nice fluffy there was like 5 inches of nice fluffy snow. And then this like grainy, sleety stuff mixed in. So actually Karen went out in the yard and measured 8 inches of snow at one point. And then like an hour later, after it snowed like crazy, she went out and measured again. It was 8.5 inches. So it's like, what's going on out here? It's been snowing like crazy and for well over an hour, and there's only an extra half inch. This much denser version of the snow started falling, and it was like squeezing down on the fluffier stuff. And actually, it seemed like there was very little accumulating, but it was definitely accumulating. We ended up with this really dense, like 11-inch snowfall.
Wow.
So I'm not going to be finding any mushrooms for a while. 4 degrees below zero on my front porch this morning. It's supposed to be colder tonight.
Yeah.
Yeah.
We're in the doldrums of winter here. So
Yeah, well, I just go out my skis,